afternoon, everyone. My name is Andrius Jonas Dagilis, and I am here today to talk to you a little bit about chromosomal inversions and how chromosomal inversions associated with mating preference evolve. So before I start on anything really big, I'd first like to remind you of what actually a chromosomal inversion is. So these are very common types of mutations where you have a segment of a chromosome that gets its gene order reversed, and so suddenly the genes are not aligning the same way as they used to. Chromosomal inversions have been studied for a very long time, and they're probably one of the first classes of mutations where we could measure many different fitness effects about them, partially because you can visualize them long before you could genotype. This has led to many theories about why chromosomal inversions evolve and their possible fitness effects, but the one that's really interesting for me is their ability to suppress recombination. So in an individual heterozygous for an inversion, recombination is suppressed over the length of the inversion for several reasons. First of all, the inversion might not line up properly during meiosis. And second, even when it does line up and recombination occurs, you may end up with gametes that have unbalanced gene products. So now we have a little bit of background about inversions. Once we really reached the age of genomics, suddenly it turned out that there were far more inversions than we had seen before. One of the fascinating patterns that has come out of this is that they are quite related to speciation, especially in sympatric speciation. So there are multiple data sets that show that inversions are more common between sympatric sister species than allopatric ones. This is from a study by Dan Hooper just from last year, where he looked at continental clades of birds, which tend to be sympatric, versus island chain clades of birds, which tend to be allopatric, and found that inversions are far more common among the continental clades. The second major observation is that inversions are also associated with mating preference. My favorite example of this comes from the carrion crow hooded crow complex in Europe. These are two different species that hybridize. And here on this graph, we have a measure of divergence, FST on the y-axis, and we are looking across the genome of a crow. There are multiple outliers here, but the one that is by far the largest and contains 81 of the 82 fixed differences between the species is on a putative inversion. This inversion captures genes not only related to the coloration of the birds, but also opsin regulators that might change how females perceive their environment. If that's not convincing for mating preference, well, there are many other taxa, including Arctic ruff, anopheles, white-throated sparrows, seaweed flies, and Atlantic cod, which all show inversions that are associated in some form or another with mating preference. The big question that Mark and I wanted to ask was, how do inversions associated with mating preference evolve? So, for various reasons, we don't believe that suppressing recombination between mating preference loci should be favored in most cases. Instead, we tried to come up with the simplest possible model of how inversions that do capture some kind of mating preference locus will be favored. So I will be showing a whole bunch of figures like this where all of these little blocks are a chromosome or a segment of a chromosome, and these are representative of individuals. In our model, all of our individuals are going to be haploid, and they're going to start out their life by being cruelly killed off. So we're going to start out with some viability selection, and we're going to have two loci that are responsible for viability selection. You can think of these as Dobzhansky-Muller incompatibilities. If you have matching alleles at these loci, you're fine, you will survive. If you have a mismatch, you will tend to die off. Now that the adults have grown up, they're going to want to mate. So individuals mate with respect to some kind of assortative mating locus. We're going to call it M, and we're going to parameterize how much they assort with similar individuals with H. So with probability H, you hybridize and you mate with someone who doesn't match your allele at M. With probability 1 minus H, you're going to mate with individuals who are the same. If H is 0, there are no hybrids. That means everyone is going to mate with everyone who looks exactly like them. When H is a half, this allele essentially does nothing, and you're mating completely at random with respect to the locus. Individuals pair up, you now have a mating pair. The mating pair is going to undergo recombination to produce the next generation. For the sake of this super simple toy model, we're going to stick our assortative mating locus smack dab in the middle of the two viability loci. All right, I've talked a lot about little haploid populations and nothing at all about inversions. So where does the inversion come in? Well, the inversion captures one of the assortative mating loci as well as one of the viability loci. What this does, especially while the inversion is rare, it will only be found in heterozygous pairings, so it will essentially suppress recombination between those two loci to zero. We will see no recombination between those two loci. All right, we developed a nice analytical math model and got some beautiful results out of it, but what I would really like to do is have you guys leave with a bit of intuition of why this model suggests that inversions are favored. So before I get to any of the math, I'll give you some idea of the fitness effects that the inversion is getting in order to be selected for. So first of all, when you don't have an inversion, disassortative matings in the population, whenever there's recombination, you will have offspring that are unfit and unviable. So if you mate disassortatively, you're going to have unfit offspring. In the case of the inversion, just because it suppresses recombination over some length of the chromosome, you might have far fewer offspring who are unfit, and so you're just going to have higher fitness because some of your offspring will do better. On the other hand, the far more important effect comes from the fact that even if you have totally perfectly fit individuals that are mating assortatively, if they have the wrong, quote unquote, assortative mating allele, they will, if they recombine, produce unfit offspring. On the other hand, the inversion essentially links one of the viability loci with an assortative mating locus, and so it will always be found with a correct copy of that assortative mating locus. So finally, let's look at a little bit of math. I'm actually going to hide most of it away behind the screen. So even in this super simple toy model, the selection for the inversion depends on 3 different parameters, which are viability selection against the hybrids, hybrids here being individuals with mismatched viability alleles, the strength of assortment, and the linkage map. There's a couple of nice things we can take away from this model. First of all, selection for the inversion is always positive if there are some hybrids. So here on the x-axis, we have the rate of hybridization. Again, if it's 0, you assort perfectly, and you're only going to mate with individuals who have the same allele. If it's a half, you're mating essentially at random. And on the y-axis, we have selection for the inversion. As hybridization goes to 0, the inversion is not selected for. In all other circumstances, there's positive selection for it. All right, how does the linkage map affect this? Well, as the loci become more and more closely linked, here represented by the R values decreasing, the strength of selection for the inversion decreases. This should make intuitive sense from what I've told you so far, because as I said, one of the main effects of the inversion is suppressing recombination. If there's no recombination happening, the inversion can't be selected for. The limiting case here is if the recombination rate is 0, so the loci are all perfectly linked, there can be no hybrids, and so there can be no selection for an inversion that captures them. All right, how does the inversion, how does selection for the inversion change with viability selection against hybrids? Well, when hybrids are, S is here the relative viability of hybrids. So as S goes to 1, hybrids do perfectly well. As S goes to 0, hybrids all completely die off. As S increases, selection for the inversion again drops because hybrids are doing much better, so there's less need for selection of an inversion. Again, the limiting case is when S is 1, you're going to have absolutely no selection for the inversion. However, under all other circumstances, the inversion is selected for, at least a little bit. So the inversion is favored under varied scenarios. And that's kind of our biggest takeaway from this. That an inversion that captures an assortative mating locus together with at least one viability locus is going to be favored under very varied scenarios. We wanted to go away from the toy model, though, and get a little bit more complex results. So we started looking at other cases. First, we looked at diploid populations rather than haploid ones, and we found again that an analogous inversion is favored. We then looked at a model of female preference male trait. In this case, you have rather than assortative mating locus, you have a female preference trait locus, a female preference locus, and a male trait locus. And if an inversion captures either one of those loci together with a viability locus, again, it's selected for under wide circumstances. Then we looked at multiple pairs of viability loci. What if there are many different DMIs along your genome and you capture some proportion of them? Again, this inversion is selected for. And lastly, we looked at complex incompatibilities, which involve more than 2 loci. And you capture even a single one of those loci as well as some sort of mating locus. The inversion is once again favored. So the big takeaway from this is And yeah, if you want to see all these results, the paper should be out sometime soon, and you can look at the actual map. But in conclusion, we find that inversions that capture loci associated with mating preference and at least one locus involved with viability are selected for under a wide range of circumstances. This provides a novel hypothesis for the association of inversions and mating preference in a wide range of taxa. Before, we probably could have said that in at least between species inversions, those might have had something to do with pre-mating isolation. But this suggests that even when you're looking within a species, inversions may be capturing not only mating loci, but also some kind of viability loci. All right, as usual, I have raced through my talk, but I'd like to thank the Prezygotic lab and open the floor for questions.
Plenty of time for questions.
Can you give an example within species where you have this sort of interactions and how that evolved?
So the best example is probably in the Arctic rough. So there, the inversion, or there are actually multiple inversions, but you have 3 different male morphs of the Arctic rough, and there are no fitness effects between those morphs, and they're completely controlled by inversions. Those morphs are have entirely different sexual strategies, so they're also clearly linked to mating preference in females.
What if the mate choice trait was an NHC type where you got a disassorted preference? Does it change any of your conclusion there?
It does. So I completely glossed over the fact that that H parameter that I kept talking about, I stop at a half, but it can go beyond a half, and if it's larger than a half, you have disassorted. In the case of these simple models, it actually suggests the inversion is not favored, but that's because we built them around the fact that we're looking at assortative mating. We probably have to look at a more complex case, and we haven't done that.
What if you have 2 mate preference loci and like 1 viability locus?
And both are captured by the inversion,
Or like 1 mate preference loci is captured by the inversion and 1 viability locus.
Yeah, it gets complicated. So we briefly looked at what happens when you have multiple assortative mating loci, and the general justice selection for the inversion is stronger, but we haven't looked at what happens when you have female mating preference loci, that there are many of them, so I don't know in that case.
Any other questions? If not, thank you for coming to your reproductive isolation.